Wait till you're announced. We've not yet lost all our graces. The hands will stay in chains. Look upon your greatness that you'll send the call out, 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 send the call out.
So we'll be starting with the lytic cycle as seen here. And so the lytic cycle is one type of the two ways viruses can replicate. So here we have the host cell DNA in the blue, and this is a phage, which first step is attachment to the host cell, and here we have the virus DNA in the green. And so after that, what happens is injection, and so it injects the virus DNA into the cell, which is what we see in green. And what happens in the lytic cycle is that the virus DNA basically breaks apart the cell's DNA, the host cell's DNA, and it takes over the machinery of the cell. And what it'll do is it'll start making the proteins for the virus out of the cell's machinery. And so that's what we see here. We have the different parts of the phage being produced here. And a phage is just one type of a structure of virus. There's many different kinds, but this is just the kind I'm showing here. And so again, we have the viruses in the head of the phage. And then after all the parts have been translated, after that we see assembly, which is where all the different parts of the phage will be put together to make the virus complete. And so after this, the lytic cycle, the root of it comes from lyse, which means to break apart. And so the lytic cycle, once it's done replicating all of the viruses, it breaks apart the cell and kills the host cell, and then all of the made viruses go out to infect other cells. So that leaves the host cell sad because it's died, but the virus is really happy because, hello, we made more. So yeah, that's basically the lytic cycle. After that, we have the lysogenic cycle, which is a different way that viruses spread their DNA. And so again, we have our phage and our attaching to the host cell. The blue is again our host cell DNA, insertion of the virus DNA, and the green. And so in this case, it doesn't actually break apart the host cell's DNA. What it does is it combines into the DNA. And when I say combine, I mean it actually integrates itself into the host cell's DNA. And so when the host cell gets ready to replicate, it's replicating not only its original DNA, but the DNA of the phage too. And so this type of DNA, where it's a mix of both the host cell and the virus cell DNA, is called a prophage. Yep. And so this just makes sure that a copy of the virus DNA is transmitted to each daughter cell as the cells are replicated. So it's kind of almost quote-unquote hiding in the cell until it wants to go into the lytic stage, where it'll produce more of the bacteria.
Since the return from her stay on the moon, she listens like spring, she talks like June. Hey, hey.
So what I'm drawing now is the rabies virus. And so what the rabies virus is it's an RNA type of virus. And so it's single-stranded. And it's shaped like a bullet, as you can see here. And the outside is covered in these glycoproteins, which just encase the virus. And then what I'm drawing here is the actual RNA. It's ribonucleoprotein, so it's the RNA which is being encased in a protein. Yeah, so yeah, the rabies virus infects the cell the same as most viruses. It goes through adsorption, which is where the membrane connects to the host cell, and so usually this is thought of to be because of receptors, but it hasn't been fully proven, but that's one of the main theories, that there's receptors on the outside of the host cell that is what the virus attaches to. But it attaches through adsorption. After that, the virus is taken in through endocytosis. So endocytosis is where the cell takes things from outside of the cell and gulps, quote unquote, or eats them and brings them into the cell. And then once that happens, the virus enters the cell's cytoplasm. Yep, and then next, since it's an RNA virus, it actually needs to be transcribed. Because it's negative stranded, it needs to be transcribed into the right code. And then once it's transcribed, it's then translated into all the proteins that make up the virus.
Whoops, that was kind of a crappy circle thing. Okay, anyway, this takes place like the Lytic cycle because what happens is the RNA is taking over the cell's machinery again, and it's not just hiding itself in the DNA to be passed on to daughter cells. It's actually using the cell's processes to create proteins and make more of itself. So this is changing the host cell's DNA because it is literally infiltrating the cell and giving it new instructions to create more of the rabies virus to go and infect more cells. And so what you see happening here is all the parts are coming together once the proteins were translated, and this is the final stage, which is called budding, where it's going to go out and infiltrate all the other cells and do the exact same thing over again. Okay, so RNA viruses are especially bad, not because it's just a virus, but because mutations are something that happens more frequently. In RNA viruses, they don't have any repair system like a DNA does, and so mutations are going to happen much more frequently. Viruses replicate at very high speeds and they produce rapidly, and so the more you have producing, the more chance there is to have a mutation, and then when that mutation goes off and starts to replicate itself, that's when you start to see the different evolutions of different strains. So this brings me on to my next point, because in 2009 in northern Arizona, National Geographic did a story on this rabies strain that has mutated and became something much more serious than the common rabies strain. And what was happening was that the strain appeared to have mutated so that foxes and skunks, which were being infected, were now able to pass the virus on to their kin. So not just through biting or scratching infection-wise, but through socialization.
And this doesn't just happen. You don't just wake up one day and say, oh, hey, look, here's a new strain of rabies. It's something that has evolved and mutated and changed along the way from what was once a different strand of rabies that had mutated in an animal and then was passed along and mutated more and has turned into this. And so that's one of the problems with the viruses, is that since they evolve so quickly and are replicated so efficiently, this is why mutations are something that is common in a virus.
of love.